Hello everyone. We know that plants they respond to various stimuli, but how? Come on, let us try to inculcate an understanding into this interesting phenomena. How a plant or an organism it responds to a signal that it receives. This entire process, starting from the signal resulting in the generation of a response to that stimuli is studied in this phenomena, the physiological phenomena of signal transduction. Now, signal transduction, it is basically a cascade of biochemical reactions which takes place inside the cell, and because of this series, cascade means at every point there are multiple factors, multiple components that are involved from the point of reception of a signal by the cell to the generation of the response. And it is this biochemical reaction that leads to the transduction of a particular signal within the cell. That means when a cell, first step of signal transduction will be reception of the signal by the cell, and the molecule that receives the signal will be the receptor. Then it will stimulate a series of different molecules which are responsible for generating the response to this signal. And as we all know, as we observe in the nature around us, this response can be multiple response. That means a single signal may induce multiple response in a system. Now, cells they can be affected by the molecules, by the signal molecules that they generate by themselves. This system is the autocrine signals. Autocrine signals means the cells which produce these signals, receptors for these signals are present on that very same cell. Second thing may be that one type of cells they produce the signals, and the receptor for that particular signal is present in some other cell. So this type of system is the paracrine system. Now if a cell do not have got the receptor for a particular signal, it will not be affected by that particular signal. Another condition may be that these signals they do not affect the cells that lie in the vicinity. Instead, they are carried over to different other parts of the body. And this type of signals, they are basically the hormones. And here lies the difference between the hormone and an enzyme, because hormones they are basically the signal molecules, and enzymes they are the biocatalysts. So this process of signal transduction may be either direct or it may be of indirect type. By direct signal transduction, we mean to say that when a signal it binds to the receptor molecule, directly the response is generated. Like in this case, when the signal molecule, it binds to this receptor, because of this binding, there's opening up of this channel, ion channel, and there's movement or transportation of these ions through this membrane. So this type of signal transduction, signal is here and immediately the response is generated without the mediation of a series of biochemical reactions. So this type of signal transduction is the direct signal transduction. However, in case of indirect signal transduction, when the receptor, it receives the signal, it induces the message to other molecules, other messengers. The receptor, it will be the primary messenger because it is receiving the signal, and in turn it will generate other molecules, other secondary messengers, and these secondary messengers or second messengers, they are responsible for inducing various cellular responses. So in this type, the signal transduction, it is mediated by primary effectors, the secondary effectors, and the second messengers. So this type of signal transduction is the indirect signal transduction. So here the primary messengers, they receive the signal and then they induce the generation of second messengers. So this is the indirect pathway. Now the various components of signal transduction. As we have just got an idea, they are the primary messengers, which are generally known as the receptors. Then there are the effector molecules, molecules which are affected when a signal is received by the primary messenger or the receptor. They are the primary effectors. These primary effectors, they induce the formation of some other biomolecules which are known as the second messengers. And these second messengers in turn, they affect some other protein molecules, because as we had said in the beginning, when we had defined signal transduction, we had used the word cascade. Cascade, basically it refers to a waterfall in which each and every rock that comes in the path of the falling water in the stream, it enhances the flow of that water. So here also the signal is flowing down, and these second messengers, they affect some other protein molecules, some other effectors which are known as the secondary effectors, and these secondary effectors in turn, they affect or they activate the third messengers. So now we'll try to inculcate or develop an understanding of these various components of signal transduction. Receptors, the messengers, the primary messengers which receives the signal. So they can be broadly categorized into two types. One, which are present on the cell membrane itself, and the other which are present within the cell membrane, that is intracellular receptors. These intracellular receptors, they may be present in the cytoplasm, or they may be present even within the nucleus. These cell surface receptors, generally they are the glycoproteins, while within the cell intracellular they may be various types of proteins, or when present within the nucleus they may be the DNA binding proteins, which directly are affected by the presence of a signal. So as a general rule, if the signal is a small non-polar molecule and it is able to enter the cell via the cell membrane, it may be a means lipophilic molecule, or it may be a small non-polar signal. So in that case, the receptors, they will be present within the cytoplasm, intracellular receptors, because signal, it directly is permitted to pass through, to traverse through this membrane of the cell. However, if this signal molecule is a polar molecule, it is large enough so that it is unable to pass through this cell membrane. In that case, one has to have a receptor which receives it on the external surface of the cell membrane, the bilayer, and then transmit the signal on the inner face of this bilayer, that is the cell membrane. So these receptors, the membrane receptors, they are basically different types of proteins which are embedded within the lipoproteinaceous membrane, cell membrane. They are the integral proteins, the integrated proteins. So when this receptor, it receives the first signal molecule, receptor, that is the first messenger, it will affect certain protein molecules, and the protein molecules which are affected by this primary messengers, by these first messengers, they are known as the primary effectors. Now these primary effectors, these proteins, they generate certain other molecules, biomolecules, which are known as the second messengers. And these second messengers, in turn, they will affect different category of proteins within the cell system. So the category of proteins which are affected by these second messengers, they will be the secondary effectors. So in this manner, the effect of the signal present on the external side of the cell, it is transduced within the cell system so that the response, it may be generated. These second messengers, now what is the need of these second messengers? What is the significance of the second messengers? We say that even a speck of signal, signal means something, the quantity of which is just sufficient. So that signal, it induces, it generates multiple responses. Its effect is amplified. So how is it possible? Basically, it is the second messengers which are responsible for the amplification of the signal received by the cell. And multiple effectors, they may be affected. The second effect secondary effectors. They may be affected by these second messengers. Now, there are so many molecules of second messengers that are produced in response to the signal which affects the primary effector. So this primary effector, it produces multiple molecules of these second messengers, and all these molecules, they diffuse out within the cell system, producing effects, different type of effects on different biomolecules. So they have multiple effectors. They are affected by these second messengers. Now, what happens when such type of amplification takes place, or when multiple effectors, they are affected by these small diffusible molecules within the cell system? They are a type of control channel, because only those cell systems will be activated which are affected by that particular category or type of second messenger. Then these second messengers, they not only affect various molecules, but at times they even alter the properties of various primary messengers. And in turn, that means when they affect the second messengers, secondary effectors, these secondary effectors, they in turn may produce the third messengers, and these third messengers, they are responsible for regulation of gene expression. So these second messengers, they carry the message, or rather they are the cascading biomolecules between the primary effector and the secondary effectors of the cell system. These second messengers, they may be of varied types, because they have to accomplish a plethora of uh, activities, a wide range of activities within the cell system. So there may be different types of ions, for example, the calcium ions. They may be lipids, derivatives of various lipids, like the DG and the ceramides. Or they may be carbohydrate derivatives, like IP3s. There may be various nucleotides, like the cyclic AMPs and the GMPs. Or they may be proteinaceous in nature, like the RAS, the JFK, and the RAF proteins. So, these are some of the examples of the various second messengers produced within any cell system. Now, a single primary effector may induce the formation of more than one type of second messengers, and thus not only this amplification of this signal, or the effect of the signal, but in diverse directions, because we know that on receiving a signal, some of the effects or some of the response of the cell system, they are immediate response, and there are certain others that are the late response, that are uh, that involves the formation of some new messenger RNAs, that involves the nuclei, the DNA, the genes of that cell, and the response is generated after some gap of time. So all this is possible because multiple types of second messengers, they are involved, and each second messenger, it takes its own route, to generate a particular response. So here is a simplified model. The stimuli, it is received by the receptor, which may be present on the cell surface or which may be intracellular within the cytoplasm or within the nucleus. These in turn may have a direct signal transduction response, or there may be an indirect signal transduction response. In case of an indirect signal transduction response, there is the primary effector proteins which are affected by the formation, by the joining together of this receptor with this ligand, the stimuli molecule. And these in turn result in the formation of various second messengers. These second messengers, they activate and affect various second effectors or the secondary effectors, and these secondary effectors, which are generally enzyme molecules like the kinase, the phosphatase, they get activated, they amplify the response all the more, and in turn they may affect the third messengers, various transcription factor proteins, which carry the message from the cytoplasm into the nuclear membrane, where they help in the transcription of a particular gene segment, because we know that in order to initiate the transcription of a particular gene, a specific transcription factor, it has to bind to the promoter region of that gene, so that RNA polymerase may start its activity on that point. So this is how signal transduction, it influences the gene expression via the third messengers. So these third messengers, they are basically the molecules which transmit message from outside the nucleus to the inside, or vice versa, that is from inside the nucleus to the outside. That is, they traverse the nuclear membrane. They are basically the DNA binding proteins. So if we see this process of this interesting process, I always consider signal transduction as one of the very interesting phenomena of physiology, of cell physiology. So it has got three steps in which signal transduction takes place. First is the reception of the signal, then its transduction within the cell system by means of different types of biomolecules, the effector molecules, the second messengers, and the third messengers. And they induce different responses which may involve the gene expression, or which may be generated directly without the involvement of the gene cells of the particular cell. So there's production of this response as a result of this transduction of the signal received by the receptor. So I hope this video has introduced you to a beautiful physiological phenomena that will open up your vision to see the nature around you. Thank you.